Welcome to this session of Pharmacokinetics, a brief overview. Myself, Khandekar Hassan Reza, M.Pharm, Assistant Professor, Bengal School of Technology, will discuss on the topic of pharmacokinetics in brief from the genesis to its application. Our discussion will rely on the basics which are very essential for the learners on this topic. Let us start from the genesis. The genesis of pharmacokinetics goes back a century. The first work related to pharmacokinetics was done in 1932 by Wildmark, who studied the time course change of alcohol in the blood. It was seen that it was following zero order kinetics, and it is a well established phenomenon which is even followed today. Later on, in 1933, Walter Gellhahn proposed that intravenously administered drugs are in function to the time. This is what which gave rise to the correlation of drug concentration with the time curve. Later on, in 1937, Torsten Theorell, a Swedish physiologist and biophysicist, proposed from his major contribution with his famous manuscript Kinetics of Distribution of Drug Substances Administered to the Body. Torsten Theorell, also known as the father of pharmacokinetics, for his extensive work which led to the foundation of modern pharmacokinetics. Later, it was in 1953 that F.H. Dost, a German pharmacologist, coined the word pharmacokinetics, which appeared in his book known as Blood Levels in English. So this is how it led to the time course role for the subject. There were many other contributions made by other scholars, but these are the preliminary contributions which brought a change or a massive upliftment of the subject. Going inside pharmacokinetics, it is well known that it is a composite process of absorption, distribution, metabolism, and excretion process. Whatever the formulations may be herbal or allopathic medicine that are taken in undergoes the process of absorption followed by the disposition. The disposition includes your distribution, metabolism, and excretion. Generally, metabolism and excretion are together known as the elimination process. Well, along with the absorption followed by distribution, which distribution, metabolism, excretion, or absorption along with what we can say as the drug disposition brings about a time course change of concentration, we can see over here. So assessment of this time course change of the drug within the body is what we call as pharmacokinetics. In much more defined way, if we want to say, pharmacokinetics is a mathematical analysis of the rate process for the drug absorption and disposition, that is your distribution, metabolism and elimination process, using a definite mathematical model. Application of this pharmacokinetics for a safe and effective management of individual patients' dosage regimen is called as clinical pharmacokinetics. The concept of the pharmacokinetics will be incomplete unless we study the process by which the drug is made available to the systemic circulation in order to show the drug disposition, followed by therapy. So this is termed as biopharmaceutics. Generally, when we discuss about pharmacokinetics, the biopharmaceutics term comes in coincidence with the pharmacokinetics terminology, because it is the process which enables the existence of pharmacokinetics. We know that there are different factors like physiochemical factors, physiological factors or pharmaceutical factors which essentially required for a formulation development. Your formulation development results in the formation of pharmaceuticals. These pharmaceuticals later goes inside the body and shows the various pharmacokinetic activity. Well, considering these three important factors leads to the concept of a science that enables the drug to be presented to the systemic circulation and enables the pharmacodynamics, that is called as biopharmaceutics. Ultimately, together along with pharmacokinetics and pharmacodynamics with the help of biopharmaceutical concept is ultimately aimed at getting a designed drug therapy. So, the major aim of pharmacokinetics along with biopharmaceutics is to determine and optimize drug therapy, and thus it is a very important concept to be considered while designing and administration of a drug. Here I show the types of bioavailability obtained upon the types of the dosage form. Generally, here I have shown three types of curve, and bioavailability actually means the rate and extent of the drug which is present in the systemic circulation. This can be obtained by sampling the drug at various time intervals and analyzing the sample. This gives a curve with concentration compared to the time. The curves which we obtain, there are three types which has been shown over here. The first one you can see that is showing an exponential decrease. This is for IV bolus. Considering the IV bolus, it shows a maximum bioavailability on administration followed by its elimination process. On the other hand, when we go for the green curve, we can see there is a comparative increase of the concentration followed by elimination. This is what we have for IV infusion. The IV infusion generally starts with a zero order kinetics and upon reaching the steady state concentration it is maintained. After taking away the drug source, that is the infusion system, it shows an exponential decrease as a part of elimination. Whereas for your extravascular administration, it generally has a absorption process followed by a elimination process. Generally, when we study the pharmacokinetics, it is being evaluated by the various parameters known as pharmacokinetic parameters. The various pharmacokinetic parameters which actually helps to assess the rate and extent of the drug in the systemic circulation are enlisted below. They are as shown ADME. You can see over here it is showing absorption, distribution, metabolism and elimination. Here what we can see generally is absorption related pharmacokinetic parameters involve Ka, that is absorption rate constant, K0, absorption rate constant 0 order. Ka generally we use it as for the first order rate constants. Generally for immediate drug release we use this rate constant. For K0, this is a rate constant we generally use actually for IV infusions. Also this is used for assessing the drug release in the case of controlled release drug delivery. F suggests the fraction of the dose that is absorbed. X0 generally is denoted as the dose of the drug which is administered. F is the fraction of dose that is absorbed, suggests a fraction of the total dose gets wasted due to various enzymatic or pre-systemic metabolism. D suggests the distribution of the drug. M, that is your metabolism. We have Km, that is the rate of metabolism, rate constant for your metabolism. Elimination generally involves clearance. Excretion generally involves clearance. Ke, that is the rate of excretion. K capital E, that is the elimination rate constant, which is a combination of excretion rate and metabolism rate. And lastly is the half-life or elimination half-life or T half. So this is the categorical division of various pharmacokinetic parameters which are used to assess the pharmacokinetics of a drug in the body. Now these parameters, if we further assess the parameters, it can be classified in two ways. One, which is any model independent or non-compartmental analyzed parameters. This includes Cmax, Tmax, AUC. The advantage of these parameters is that we can directly assess the parameter from the concentration time curve. There is no need to further assessment or calculation or derivation to assess or judge the presence or of these parameters in a system. Whereas other parameters like volume of distribution, clearance, intercompartmental clearance, absorption rate constants, bioavailability and the clearance generally needs to be calculated based on a particular model. So this is one way of classifying your parameter. Considering your model, considering both the parameters, model dependent as well as model independent, both are essential and required for pharmacokinetic analysis. The other way of classifying the parameters is number one as a primary pharmacokinetic parameters and secondary pharmacokinetic parameters. Now, when we are discussing this interrelation of pharmacokinetic parameters, let me say that the clearance and volume of distribution considered as primary pharmacokinetic parameter, as they are independent to denote a single physiological process. That is, volume of distribution symbolizes the distribution process which is taking place in the body, whereas the clearance generally this defines your elimination process that takes place in the body. And the change of these pharmacokinetic parameters, specifically your volume of distribution and the clearance, does not affect the change of the other. The parameters like T half, rate constant and bioavailability does not denote a separate physiological process, and most of the time it results in a composite process that mixes multiple physiological process parameters. That is, most of the times they are generally involved on the primary parameters. The primary parameters generally influences the existence of the secondary parameters. Now, as I told you, the pharmacokinetic models is very essential tool to assess the pharmacokinetic considerations. Generally, raw data is obtained as plasma concentration time study. This plasma concentration time study is put inside a mathematical tool. Generally, we call it as a pharmacokinetic model. And through the assessment of the pharmacokinetic model, we can determine the various pharmacokinetic parameters that assess the pharmacokinetics of a particular drug in our body. Generally, the different types of pharmacokinetic parameters which are present are compartmental model, non-compartmental model and physiological based pharmacokinetic model. These are the main, you can say the main models, important models which are mostly used for assessing pharmacokinetic parameters. When we say a compartmental model, you can see the body is considered as a homogeneous unit boxes or compartments in which there is a equilibrium movement of the drug which is being established. Generally, they are of two types, mammillary model and quaternary model. But in our physiological process, we consider the mammillary model, in which comprises of a central compartment which is having a high vasculature, through which the drug gets equilibrium with the secondary and tertiary compartments in the time course. So this is the most common compartmental model and the oldest model which is commonly used. The other model is called as non-compartmental model. It requires a linear pharmacokinetic assumption and is based on static moment theory. The other model, which is physiological based pharmacokinetic model, is little realistic picturization of the drug disposition and the movement. The disposition highlights the relative alteration of the perfusion rate of the blood to the different organs. And the number of organs to be considered generally depends upon the rate and extent of the drug disposition that is taking place based on the perfusion rate of the blood. So this is the most realistic and advanced model which is present for assessing pharmacokinetic parameters. Let us go for some concepts and assumptions that we frequently make while doing the pharmacokinetic assessment. First one, the rate process generally follows a first order kinetic. This is a preliminary assumption that we make. The total physiological process which is taking place in our body follows a first order rate kinetics. Secondly, there are certain rate kinetic process which are initially follows a first order followed by a saturation to a zero order kinetics. It generally occurs to a rate limited processes, and it is being explained by Michaelis-Menten equation. The first order rate process symbolizes the exponentiality. Generally, it is said as mono-exponential if one first order rate process is involved, or bi-exponential if two first order rate processes is involved. Like in IV bolus, it involves only elimination process, so it is mono-exponential form. Whereas for extravascular administration generally involves two rate processes, that is absorption and elimination, and hence it can be bi-exponential. In the other case, the degree of drug presentation is the first thing which we consider for assessing the pharmacokinetic study. Generally denotes the difference at which the drug goes inside the body, that is the rate in process, and the rate at which it comes out of the body, that is the rate out process. Next we go for applications. First application is biopharmaceutical consideration for designing formulations suitable to a definite route, making the drug bioavailable with a defined pharmacokinetics. Pharmacokinetic study makes us or helps us to design a drug of a suitable rate such that it can reach to a particular site at a particular rate and can show its drug activity. Nextly, this pharmacokinetic consideration is very vital in the regulatory process, specifically for submission of IND application. It is also useful for submission of dossiers to other countries for a drug approval. It is also helpful for bioavailability and bioequivalence studies, and also these pharmacokinetic considerations of a drug helps us in the alteration of dosage regimen of individuals in case of disease or altered state. So these are the primary places where we can use the concept of pharmacokinetics in the study. Okay, these are some of my references which I have used along with my personal experience regarding the subject has been discussed till now. If you are interested